So moving on from the astrocytes to the oligodendrocytes, or oligodendroglia, these are important in forming the myelin sheaths around axons in the central nervous system. The Schwann cell does kind of the same thing in the peripheral nervous system, but again, remember, one Schwann cell per neuron and one oligodendroglial cell per many neurons in the brain. Loss of oligodendroglia is often associated with what are called the demyelinating disorders, or various disorders that result in the actual loss of the myelin sheath. And these diseases would include things like multiple sclerosis or amyotrophic lateral sclerosis, or ALS, or Lou Gehrig's disease. Those actually involve loss of the oligodendroglia. In fact, within recent years, so just the last couple of years, ALS, which is a horrible disease that results in loss of all muscular function, but really no compromised cognitive function, a lot of treatments are being directed toward trying to maintain the health of the oligodendroglial cells. So that's an important clue as to what might be causing some of the damage in ALS. And we also see a specific type of neuropathology in the oligodendroglia of patients with multiple system atrophy called the argyrophilic cytoplasmic inclusion. And multiple system atrophy is actually a whole host of diseases that we often refer to as Parkinson's plus. They have many features of a Parkinson's patient, but they have additional symptoms depending on the area of damage. One horrible type of disease is called Oconto cerebellar atrophy. And again, the name of the atrophy tells you where the atrophy is occurring: the cerebellum, the pons, and the olive. Shy-Drager disease is yet another Parkinson's plus type of multiple system atrophy that is among the more common multiple system atrophies. But when they're testing for that, they look at a specific sample of the oligodendrocytes to see whether they can detect these argyrophilic cytoplasmic inclusions, and that would be diagnostic of the multiple system atrophy. And this is again an example of the oligodendrocyte, one oligodendrocyte for many myelinated axons. Notice, though, that with the myelin sheath, there's a little gap of bare neuron, and that little gap of bare neuron is very important for the action potential to propagate. That's called the node of Ranvier. And so we'll talk more about the role of the node of Ranvier in generating or maintaining an action potential. So again, this myelin sheath is really helpful for neurons to enhance the conduction of the neural signal. And it prevents neurons from also being sent to other nearby neurons. So it's sort of like a insulation around an electrical wire, rather than just having the bare wire that could, if you touch it anywhere, it could, electrocute you. If it is covered, then it protects that signal, that electrical signal, from being sent out. And this is a Schwann cell. We have our node of Ranvier again, also in the Schwann, in the peripheral nervous system, but one Schwann cell for one myelinated axon. And this is yet another diagram of the formation of myelin. Here is the oligodendrocyte that sends out little appendages and starts to wrap around the axon there's several layers, and it does that for many of the sites that the oligodendrocyte is close to. And in the peripheral nervous system, we have the nucleus of the Schwann cell that's represented by this little dot down here, and it's again surrounding the axon, and it keeps going around and around the axon, just one Schwann cell per axon. Yeah.
So question, I know myelination is a process that happens during development of the brain
Mm-hmm.
as you age, but does it happen in adulthood as well?
Yeah, absolutely.
It continues to.
It continues to up until about age 25.
Mm-hmm.
So if you're under 25, your brain is still involved in the myelination process. So then we have the glial cell, or microglia cells, and these are the smallest of all the glial cells, hence their name, microglia. And they're the brain's innate immune system that I mentioned before, and they're mostly found in gray matter. And you can see right here in the middle, this is what they normally look like under sort of physiological conditions. They're in what's called a ramified or resting state. And they very much resemble a star-shaped astrocyte because they have these many appendages with one sort of center part. These little thin processes sort of extend in all directions, just like astrocytes. And in the resting form, each of these microglia is sort of like on the lookout for any kind of invasion of the brain due to an infection or damage, physical damage. They become activated in response to mechanical, chemical, or immunologic injury, and the cell actually transforms from this sort of skinny, processed, star-shaped form to an amoeboid type of form. As you see down here, it's much sort of fatter in the middle, and the processes get much thicker, and it becomes motile. It actually moves throughout the brain to kind of clean up all the injury that surrounds it. And so it sort of wraps around the damaged area and kind of digests it or phagocytizes it. And so this phagocytation or cleanup crew in the brain releases vasoactive and neuroactive cytokines. Cytokine is a protein that regulates immune system functioning or stimulates immune system functioning. And the microglia also release pro and anti-inflammatory substances. So they try to stimulate inflammation in some areas and try to inhibit inflammation in other areas, depending on need. And more recently, the microglial cells have been focused on in Alzheimer's disease. And in fact, I have a grant that we're working on with Cleveland Clinic, where we are looking specifically at a protein that is found within the microglia and we're trying to study how that particular protein is affected by physical activity. Physical activity produces a lot of anti-inflammatory types of benefits, so we want to see how it affects the immune system, physical activity that is, and then how physical activity affects specifically the functioning of the microglial cells through a particular gene. And then microglia is also involved in immune responses in HIV or AIDS-related dementia. So now moving from the neurons and the glial cells and the synapses into the neuronal membrane, this is an interesting phenomenon whereby all neuronal membranes have a resting membrane potential that's governed by the relative concentrations of various ions inside and outside the cell. And so each lipid molecule has a head, which is hydrophilic or likes water, and a tail, which is hydrophobic or doesn't like water. And the tails orient away from the water, forming a double-layered membrane, as you see here. And so molecules like water, oxygen, and carbon dioxide can pass freely through this membrane. And again, these are just little molecules that are kind of all bumping up against each other. And then other molecules or ions can only pass through protein channels in the membrane under certain conditions. And so we say that the membrane is selectively permeable. It only allows certain ions to pass through under certain conditions. And that selective permeability causes the membrane to be polarized, and that polarization means that the electrical charge is different inside and outside the cell. And so the cell membrane itself is made up of a phospholipid layer. It's a lipid and protein, and it's bathed in a saline solution, or kind of like a salt water. And what we see here is we see a high concentration of sodium and chloride on the outside of the cell. That kind of stands to reason because the outside of the cell is salt water. So a lot of sodium and chloride on the outside of the cell. The cell's being bathed in that saline or saltwater solution. On the inside of the cell, we have potassium, which can leak through a little bit, but and it wants to actually leak through because it wants to fall down a concentration gradient. There's high concentration of potassium on the inside of the cell, and wants to get across to where there's a low concentration of potassium on the outside of the cell, so that the concentration is equalized. That's the force of diffusion, trying to equalize the concentration on either side of the membrane. But because of the nature of the membrane, potassium can't get out of the cell except under certain conditions. And then we also have a lot of negatively charged anions. These are negatively charged proteins that are much too large to leave the cell under any circumstances. So because we have a lot of those inside the cell, the inside of the cell tends to have a large negative charge. Here we have also sodium. Sodium wants to follow down its force of diffusion gradient. Not only does it want to go from a higher concentration outside the cell to the lower concentration inside the cell to equalize the concentration, but it also is being sort of pushed in because the positively charged sodium is being attracted by the negative charge on the inside. So these relative forces of electrostatic pressure and diffusion are operating then to keep the certain ions outside the cell and certain ions on the inside of the cell. And so the resting membrane potential, or RMP, is just a difference in voltage between the inside and the outside of the axon membrane. As I mentioned, these anions, which are proteins and amino acids, negatively charged ions, they're on the inside of the cell, and because of their size and the lack of an ion channel to let them out, they can't leave the inside of the cell. And at rest, some potassium ions, as I mentioned, can leave the cell, causing the exterior of the nerve cell membrane to be slightly positive relative to the inside of the axon. We have, again, the force of diffusion, the movement of molecules from high to low concentration that sort of counteract each other in some cases. We have electrostatic pressure, which is the attraction of particles with opposite signs or repulsion of those with the same signs. So again, this setup kind of produces a charge, a relative difference in the voltage between the inside of the cell and the outside of the cell. So